Hello everyone and welcome to my YouTube channel Perfect Lab. In today's video, we are going to learn about selective media, which are culture media designed to allow the growth of certain microorganisms whilst inhibiting others. So in this video, we will discuss what selective media are, how they work, their components, and some common examples used in the laboratory. So let's get started. Let's start by looking at what a selective media are. So a selective media is a type of microbiological culture medium that allows the growth of certain microorganisms whilst inhibiting the growth of others. And this medium basically contains specific substances like antibiotics, dyes, salts, low pH, or other chemicals that inhibit the growth of unwanted microorganisms whilst allowing the organisms of interest to grow. The general composition of selective media. So the first component of the selective media is basal nutrient, and these nutrients basically support microbial growth. And examples of these basal nutrients are peptone and tryptone, which serves as a source of amino acids and nitrogen for the medium. We also have carbohydrates such as glucose, lactose, and mannitol, which serves as a source of energy, and agar as a solidifying agent for solid media, and beef extract or yeast extract that provides the medium with vitamins, minerals, and other growth factors, sodium chloride to maintain osmotic balance, and water as a major solvent to dissolve all these basal nutrients. And the second component of the selective media are selective agents. And basically, these agents inhibit some organisms whilst allowing the growth of others on the same medium. And examples of these selective agents are bile salt, antibiotics, dyes, high salt concentration, low pH or acidic conditions, and chemicals. So let's look at the bile salt. So the bile salt is basically used in the preparation of MacConkey Agar to support the growth of gram-negative bacteria whilst inhibiting the growth of gram-positive bacteria. So antibiotics such as chloramphenicol, which is used in the preparation of fungal media such as Sabaros dextrose agar and potatoes dextrose agar, suppress the growth of bacteria whilst allowing the growth of fungi. And we have dyes like crystal violet, eosin, methylene blue, which are used in the preparation of medium to inhibit certain bacterial growth. And we have high salt concentration, and this is basically 7.5% sodium chloride, which is used in the preparation of Mannitol Salt Agar to select for salt-tolerant organisms like Staphylococcus. And the low pH, which creates an acidic environment or conditions, create an environment that favors fungi growth whilst inhibiting most bacterial growth. And we have chemicals like sodium azide, which are added to certain selective media to inhibit the growth of many gram-negative bacteria to allow the selective isolation of gram-positive organisms such as enterococci and streptococci. Examples of selective media. And in the examples of the selective media, we will examine some of the most commonly used selective media in the microbiology laboratory, and for each medium we will identify its key selective components, the microorganisms it is designed to select or allow to grow, and the microorganisms whose growth it inhibits. We will also learn about how each selective medium can be used to differentiate the selected microorganisms based on the appearance, color, or morphology of the colonies they produce or form on each of the medium. The first selective medium we are going to look at is the MacConkey Agar. And in the MacConkey Agar, The selective components are bile salts and crystal violet, and these substances select or allow the growth of gram-negative bacteria but inhibit the growth of most gram-positive bacteria. And this medium is also used or can also be used to differentiate the selected gram-negative bacteria that are allowed to grow on into lactose fermenters and non-lactose fermenters. So those bacteria that ferment lactose, such as E. coli, produce pink or red colonies because they are able to produce acid during the fermentation process to cause a color change of the pH medium. And those that do not ferment lactose or the non-lactose fermenters, such as Salmonella, will produce a clear and colorless colonies on the medium as shown here. The second selective medium we will look at is the Mannitol Salt Agar. And in the Mannitol Salt Agar, its selective component is 7.5% sodium chloride. And this high concentration of salt allows the growth of salt-tolerant organisms like Staphylococcus species and inhibits the growth of many non-salt-tolerant bacteria. The Mannitol Salt Agar medium is also used to differentiate these salt-tolerant bacteria that are allowed to grow on it into mannitol fermenters and non-mannitol fermenters. Example, Staph aureus ferment mannitol to produce acid that turns the medium yellow, and this results in yellow colonies surrounded by yellow zones as shown in the picture here. And on the other hand, Staph epidermidis, which does not ferment mannitol, remains pink or red on the medium, with the medium not changing color. The third selective medium we will look at is the Saburo-dextrose agar. And in the Saburo-dextrose agar, the selective component is the low pH or the acidic medium created in the medium, and in some cases the addition of antibiotics. And these substances or selective components allow or select the growth of fungi but inhibit the growth of many bacteria. And this medium can also be used to differentiate yeast from mold. Example, yeast typically produce creamy and smooth colonies on this medium, whereas molds produce fuzzy and powdery colonies because of their filamentous growth. And the fourth selective medium we will look at is the eosin methylene blue agar, or in short EMB agar. So in the EMB agar, the selective components are the dyes eosin and methylene blue, and its selective component selects or allows the growth of gram-negative enteric bacteria and suppress or inhibit the growth of most gram-positive bacteria. The EMB agar can also be used to differentiate these selected gram-negative enteric bacteria into lactose fermenters and weak or non-lactose fermenters. So for example, strong lactose fermenters like E. coli will produce dark colonies with a distinctive metallic grain sheen on the medium as shown over here. So whilst non-lactose fermenters will produce colorless or transparent colonies. And the fifth and the last selective medium we will look at here is the xylylysine deoxycholate agar, or XLD agar. So in the XLD agar, the selective component is sodium deoxycholate, so which is a bile salt that inhibits the growth of many gram-positive bacteria but selects or allows the growth of Salmonella and Shigella, which are gram-negative gut bacteria. And this medium also helps to differentiate these selected bacteria based on carbohydrate fermentation and the production of hydrogen sulfide. So example, Salmonella produces red colonies with black centers because it produces hydrogen sulfide, and Shigella forms red colonies without black centers because it does not produce hydrogen sulfide. So in summary, though these media are considered as selective media, they can also be used to differentiate bacteria, as shown in the table here, where MacConkey Agar selects gram-negative bacteria and differentiates lactose fermenters and non-lactose fermenters, and the Mannitol Salt Agar selects Staphylococcus species and differentiates mannitol fermenters from non-mannitol fermenters, and the Cerberus dextrose Agar selects fungi by inhibiting many bacteria with its low pH and sometimes using antibiotics, and the EMB Agar selects gram-negative enteric bacteria and differentiates strong lactose fermenters from weak or non-lactose fermenters, and the XLD Agar selects Salmonella and Shigella and differentiates them by hydrogen sulfide production and colony forming. So thanks for watching my video, and if you found this video helpful, please like, share, and subscribe to Perfect Lab. Leave your comments or questions below, and don't forget to scan the code on your screen to follow us on WhatsApp for more laboratory updates and tutorials. See you in the next video.